fantastic to be here. My God, what an exciting place with all the AI that is going on. We just heard from a digital CEO and prioritization system for getting leads, and of course a music teacher. How awesome. So with that, I want to talk to you about the research that we do. But before I get started with that, I want to introduce the research organization that I am part of, which is CSIRO. Then I'll talk about Variant Spark, which is our mechanism for finding disease genes, and we're using the Spark, Hadoop Spark engine for that. And I want to introduce DT-Scan2, which is for genome engineering, which is a serverless architecture that is one of the genome engineering ways of prioritizing the sites that researchers want to search for. Sorry about that. We need to put in the clicker, of course. So, but before we go to that, hopefully it connects eventually. I want to talk a little bit about the trends that are in the AI space. So here I'm pulling out what PricewaterhouseCoopers thinks are the trends, and I think they're pretty close. So I'm not going to talk about each one of those trends right now. They will pop up throughout the presentation, and I talk to them in the context of where I see they fit. What I want to say, though, is that I'm not talking about all 9 of them. In fact, okay, it's not connecting. In fact, there are a couple of them that I don't really want to talk about. So the first one is that they think it's the streaming application is the next big thing, which is probably right, but not in the context of what I'm talking about here. Also, deep learning, yes, of course, it's a trend. We're not the trendsetter, so I'm not going to talk about that. And funding. Well, of course, there's oodles of funding in the AI space, but I would argue that if you have a good product, you will always find funding for that. So, CSIRO. CSIRO is Australia's government research agency. And we have 5,000 staff. Most of them are PhD researchers. We are in the top 1% of global research institutions. But what makes us really unique, yeah, I'll stay there. So what makes us really unique is that we're very collaborative. So there are 80% of the companies that think that they're going to be disrupted in the next 5 years, but only 7% of them have a plan for it. So the plan, in our mind, obviously needs to be research. You need to stay at the cutting edge. And in order to do that, you probably want to partner with someone who is actually doing research and development, which CSIRO is obviously an option for. And at CSIRO, we're very passionate about translating research into products that people can use in their everyday lives. And probably our most famous product is that we invented modern Wi-Fi. So I'm part of the eHealth Research Centre, which is Australia's digital health agency. And our Wi-Fi equivalent is a mobile health app called Toddy Hub. So it is used for when someone has a heart attack, getting them into rehabilitation. And you would think that having an adverse life event like a stroke or a heart attack will get you into better lifestyle choices. Well, it turns out you don't. It's not a good incentive. This app, though, has increased the uptake of rehabilitation by 30% and the completion by 70%, all through gaming, AI in the background. So this little app already has saved lives. So, on to our product, Variant Spark. So by 2025, 50% of the world's population will have been sequenced. And the reason for that, obviously this is trend number 1, big data. Big data is everywhere, in the genomic space, probably more so than anywhere else. Because genomic really affects the way we look the disease risk that we have and the behaviors we exhibit. So I always like to do this little exercise of, if you look at your thumb, the way that your last digit bends back, that's controlled by genetics. So if you compare your thumb to the person next to you, you can compare your bendiness of the last digit. There will be 1 in 3 people here in the room that have a super bendy last digit, so I'm not. I'm quite boring there. But yes, I can see some impressive specimens down there. So the other thing is coriander. The way you perceive the taste of coriander is also affected by your genes. And usually in a Caucasian audience, it will be 1 in 6 people that hate coriander with a passion. In South Asia, it's much lower, so it's only 1 in 12. So are there any coriander haters amongst us? Okay, 2. Very good. So obviously, this is a fun bit of what the genomics or the genes control. But obviously, there's a dark side to that as well, which is disease risk. So for example, here I'm highlighting cystic fibrosis, which is a genetic disease where 1 change of the 3 billion letters of the genome can lead to a devastating disease. So therefore, with that, it's no surprise that sequencing people and finding out their genetic risk is going to come into a healthcare system. So as such, genetics is going to be the big data discipline going forward. It will outpace traditional big data like astronomy, Twitter, and YouTube combined 3 or 4 or 5-fold. By 2025, there will be 20 exabytes produced, genomic data produced per year, which to me is marvelous on one hand, but it's quite frightening on the other hand. Because that means you have to do machine learning on 1.7 trillion data points. So here specifically I'm highlighting Project MINE, which is an international consortium to find the genetic origin of ALS, which is a motor neuron disease that Stephen Hawking has, or you might be familiar from the Ice Bucket Challenge. So in order to identify what is the genetic cause of this disease, you need to analyze 22,000 whole genomes. So this is why it's an international consortium. And 22,000 individuals, on average, between you and the person next to you, there are 2 million differences in your genome, so 3 billion letters, 2 million differences, 22,000 samples that equates to this number that I'm quoting here. And doing machine learning on that kind of data is challenging. So how to actually find disease genes? Well, it is a machine learning task, right? So you have every line here represents an individual. First, you identify what makes that individual unique. So what are these 2 million differences in those individuals? Those are the little bars that I'm showing here. Then you recruit your cases and your controls, so the people that are healthy and the people that have the disease. And all you need to do is find which ones actually separate them. Now, this sounds easy because it is too easy. That's not the reality. The reality is much more complicated than that. In fact, there's unlikely to be one location in the genome that neatly separates cases from controls. It will be more sets of things that are actually separating them. So from the little examples that I had here, I have 2 different sets of locations, 2 different sets of disease genes that work together in order to inform or cause the disease. So finding that, as I said, is a really complicated machine learning task, but it's very suitable for random forests because it can build a lot of internal models that represent the complexity of that disease. So with that, trend number 5, I would say, is artificial intelligence. Whether artificial intelligence, machine learning, whether there's a real separation or not, that remains to be discussed. So to recap that again, it's 1.7 trillion data points, and that comes from having 80 million features and 22,000 individuals. And of course, our prediction label is whether or not someone has a disease. So in order to solve this problem, we cannot really use traditional compute anymore. Certainly we can't use a desktop compute. We can't use high-performance compute because high-performance compute is really compute-intensive, or it's designed for compute-intensive tasks, ideally of tasks that have nothing to do with each other, and you can run in embarrassingly parallel applications. So, for example, weather forecast is an ideal application case for that. But for genomics and for machine learning, where the whole data set needs to inform the prediction at the same time, this paradigm doesn't work anymore. You need to have something where really the full data set can be used at the same time. And Hadoop Spark, obviously, is the ideal solution for that. So the way I think about it is that traditional high-performance compute, you have lots and lots of CPUs on there, but these CPUs are sitting in their own isolation, in their own boundaries. And if you want to develop a technology that truly parallelizes and uses them combined, that is quite an involved task. It's certainly not standardized, it's bespoke parallelization or calculating it, basically using all the CPUs at the same time is difficult. Whereas with Hadoop Spark, all of this is standardized, right? So therefore, the boundaries between the nodes really dissolve. And that makes it easier to be actually using the full spectrum of the cluster and distributing the data at the same time. So therefore, we built a new technology directly on top of Spark Core called Variant Spark. So basically, it sits alongside Spark ML or MLlib, if you're familiar with that. But the reason that we had to build something new was that Spark ML and MLlib were not catering for our use case. And specifically, our use case is wide data. So as I said, typically, the application cases for machine learning, they have many samples, but the width of the data, so how much detail you actually have per data set, is quite limited. Right? So in a typical sentiment analysis or customer analysis, there's only so much information you can collect about a user. Whereas here, we have the full genome, which means 3 billion letters. So how do typical, more traditional parallelization strategies for random forest work? So obviously, the simplest one is where each decision tree is calculated separately on their own CPU, and then the results are combined into the random forest. But that is not very efficient, and certainly for data set sizes like this, it doesn't work. So this is the first parallelization approach, which was invented by Google, and it's the Planet implementation, which basically calculates or has the parallelization a little bit more clever in that they have every sample separated or put on their own CPU. And then the results are combined in order to build each individual decision tree. Now, the problem with that is that it works really well for customer data where you have 100 features. But it does not work, it fails if you go to millions of features, because this amount does not fit into the CPU or the memory of a compute node. So therefore, Berkeley and Yahoo came along and said, well, let's just flip the problem then. Rather than fitting in a whole sample into memory, let's fit a whole feature into memory. Which sounds great. I mean, it worked for them for 10,000 features. And this is sort of what I'm showing here is that there's a nice boundary between where MLlib, which is Google's Planet implementation, works better, versus Yggdrasil, they call it, this approach works better. But the problem is that as soon as you have many samples and high-dimensional features, you're in the same pickled spot as you were before. Therefore, we came along and did a different parallelization approach, which essentially is sort of treating each cell, like samples versus features, each cell independently, and therefore combining only the ones that you actually need to compute the decision tree that you have at hand. So we apply this through every layer. So you have multiple decision trees We apply this at every layer in order to build the decision trees in parallel. So with that, obviously, you can do your standard classification, predicting disease versus control, or you can do what we call association studies, so finding the disease genes. You would call it, in the machine learning world, feature selection, where you would find the actual feature that are predictive of the label. So we implemented all of this, and obviously, as data scientists, we want to see whether it actually worked or whether it's just a nice concept. So we tested this. We tested the accuracy versus the speed. Spark ML, that's Google's Planet implementation, and Variant Spark. So Variant Spark is both in terms of the accuracy and in terms of the speed better suitable for that kind of application. And I'm not saying that the way we implemented is the perfect way and the only way. What I'm saying is we were the first with that actual problem of having wide data that needed a solution. Okay, so if you're predicting your disease status, that is akin to other application that you might have. And the features that we might have, which is here the genomes, or the mutations in the genome, they might be quite similar to things that you encounter, which would be, for example, time series data, or concatenated data, or sensor data, or log files. Like with the, as I said, with the datification of everything, data sets will grow in size because now you can automatically and continuously collect information about an individual sample, which lets the feature size grow probably without boundary. So therefore, rather than disease status, you might want to predict churn rate or occurrence of failure or attack. And rather than disease genes, you just want to know what are the predictive features. So what are actually the sensor data that can predict failure rate maybe a week, 2 weeks, a month before it actually does? Or what are the concatenated data about customer behavior that lead to churn at a point where you can actually still intervene and keep that customer on board? So therefore, this is machine learning as a service. The idea here is that you have an algorithm that is agnostic of the actual application case. You stick it into public cloud providers and make it available as a service for other people to just use this entity and apply it to their data. Tweak it, train it maybe new, but apply it to their data. So with that, let's step a little bit back and talk about the workflow patterns of data science or in machine learning. You typically start with your business problem, right? So churn rate, for example, or for us, disease gene prediction. Then you need to identify what kind of data sets will help you in order to build something, to generate a hypothesis, or at least, have a lead or showcase your ideas to your bosses. And in order to do that, you need to clean the data probably, because usually the data does not come on a silver platter, which means that you need to visualize it, you need to build statistics on it, and you need to just generally process it. And for that, the ideal way that has emerged is to use notebooks. So Jupyter Notebook, for example, where you can write a little code blurb, and underneath will be the visualization, and then once you have a different hypothesis, you can easily change it. From this initial data exploration and the hypothesis that you might have, you can build your minimal viable product. So a quick prototype to showcase to your client, for example, or to your bosses. And for that, you need to scope the technology, where to build it, like public cloud or on-premise, what kind of technology, TensorFlow, or is it a more custom approach? You build your prototype, and probably you find that all your brilliant ideas were totally rubbish when you actually put it in, when you actually make the prediction. So this is an iterative process until you have a prototype that is actually useful. The last step, of course, is once you have that prototype, you want to put it into production, because there's no, prototypes are only useful if they're actually used for something to generate value or to improve revenue. So therefore, putting your prototype into production is not a trivial task. You need to test it at scale. And obviously you need to provide some sort of endpoint that another business application can use it. So in order to follow all these 4 steps, you have a couple of options. You either can use it on-premise, like you can run it on a server, on a desktop that is sitting at your home. There I would argue that you will not get to the very end of putting it into production, because it will not necessarily be available on the cloud. It will not necessarily be as robust as it could be or should be. But it will cover most steps, and it's probably the easiest way to get started. Another option is Databricks. So Databricks is an American company that is basically a Jupyter Notebook, a managed Jupyter Notebook service, where the Jupyter Notebook is, it's actually not a Jupyter Notebook, I was wrong. It's a proprietary notebook that they wrote, but it's the same concept. So that notebook is connecting to AWS, to your AWS account, and it is perfect for this first step of curating the data, getting a feel for the data, and potentially building a minimal viable product, although that's a bit iffy, so therefore it's not just going all the way. But it certainly cannot cater for putting it into production. Whereas AWS SageMaker can, and the previous session told you all about it, so I don't need to. So with that, I would like to give you a little demo of how variant Spark works, how disease gene prediction works. The problem, though, is that with genetics, there's a privacy concern around it, right? So therefore, I can't just show you our research products, because the participants will jump up and down and will say, this is not what we signed up for. Therefore, I would like to solve another important problem, which is, are you a hipster? So with this, it's a synthetic data set that we put together, which we call the Hipster index. So it's locations in the genome that referring to certain traits that we put together in a score in order to label samples with 0, being a hipster, or 1 of not being a hipster. And then we run variant Spark on it to see whether we can reproduce the results that we put in. So with that, let's go to the Databricks notebook. Actually, I can sit down. And as I said, it's sort of little snippets where you can write your code or you can write annotations and you can execute it in the environment. So with the Hipster index, as I said, those are real locations in the genome that are associated with certain traits that we feel are hipster traits. So for example, having a monobrow, or wearing checked shirts, or having a beard, or having huge amounts of coffee consumption. I'm sure some of you can relate to that. So I'm sure some of you have some hipsterness in you. So with that, we calculate this score, which basically represents what a complex genetic disease would look like. So there will be multiple locations in the genome that interact and work together in order to cause this particular disease. So this notebook basically walks you through step by step of how to do it for your data. So the first instance, obviously, is to load the actual data. And as you can see, it's sitting on S3, an S3 bucket. Therefore, if you want to run it yourself, all you need to do is putting it in an S3 environment as well. This is also available on Azure, so this will be a blob storage as well. So with that, we load the actual data. We load the labels. So these are the, whether a sample is a hipster or not. There's some configurations. So for example, we want to build 1,000 trees on this particular data set. And then we run the analysis. And it's really just a one-liner. The other thing that is quite interesting with notebooks is that you can combine multiple languages. So typically, from a data science perspective, you have your favorite language, but it might not be the same that the next person in your data science team has. So therefore, what I'm showcasing here is that you can use different languages, your preferred language, in order to interrogate the data. For example, this is the SQL query, where we want to have the first 25 important genes or locations in the genome that are associated with being a hipster. Similarly, this is a Python example, same difference, and an R script that also visualizes the actual locations that were pulled out. So with that, let's talk about the results. Oh dear. Okay, this is tiny. So these are the actual results, all the way over there. So what I'm showing here is basically the whole genome. Every dot here represents a genetic or genomic location, 3 billion letters, as I said. And on the y-axis, I'm showing the association of this particular location with the disease. So the higher the dot, the more associated with it. And as you can see, there are the 4 locations that are standing out like sore thumbs that are associated with monobrow, fabulous hair or beards, checked shirts, and coffee consumptions. So therefore, if this was a real genetic diseases where you don't know what the actual genomic locations is, and they stand out like that, you can be pretty certain that you found biomarkers that are predictive of the disease. So with that, since this is an important problem, but it's also a universal problem, I would really like to build a community around this, where we build machine learning methods that cater for wide data, and if we can solve, incidentally some genetic diseases or cure some genetic diseases, all the better. So therefore, a lot of people come to me and say, hey, Dennis, this sounds really fascinating, but I don't have a background in research. How can I help? Or can I help? And always the answer is, yes, of course. Your background is ideal in this particular domain. And to give you some examples, Lynn Langit, for example, that you might be familiar from lynda.com. She is one of the main contributors on lynda.com. She did a performance comparison that enabled us to make the argument that SageMaker is actually the perfect way to go by showing that, or comparing Databricks to EMR and showing that the runtime difference is not, basically there is no runtime difference, which means we can use SageMaker. Similarly, the other example is Stews, which is a consulting company in Melbourne, and they ported our Scala-based API for variant Spark onto Python, because obviously this is a more accepted language in the community. So with that, let's switch gear a little bit and talk about GTScan2. So you might have heard about genome engineering, CRISPR, and that is actually probably bringing in a new golden age of medicine, because now it is possible to edit the genome of living organisms. And with that, you can imagine a world without genetic diseases, a world where the growing population can be fed. And how close we actually are to this perfect world was demonstrated by a Nature paper last year, which demonstrated that you can edit out hypertrophic cardiomyopathy, which is a heart disease, a single gene-based heart disease. You can edit that out in human embryos. And they managed to do that in 7 out of 10 cases, which is great news, except that in 3 out of 10, it did not work. And if you think of this being your unborn child, then 3 out of 10 failure rate is just not good enough. Therefore, we want to build a technology to make that work the first time, every time. And for that, because obviously we need to come in from the computational side, we're using Lanta function. And the reason we're doing that is we need to improve the speed, because with the speed, with the same budget, you can make more experiments, you can test more variables in order to identify the actual set that really works. Also, researchers might not want to, or they might want to interrogate one gene, or they might interrogate the whole genome, which is 100,000 genes. And for that, it needs to scale. So therefore, small individual tasks that need to be scalable as a web application, that is the perfect application case for Lanta. So therefore, I'm sticking here the democratization of technology in here, because this I really feel strongly about, that before, in order to make these kinds of analyses, you had to have a supercomputer, which really limited it to only the top tier research organizations. But now you can have, you can do this optimization or identify the optimal target site for your genome on the cloud, and therefore you don't have to have the infrastructure. Well, it's not the infrastructure that is limiting you from doing a good experiment. So with that, let's talk about the architecture. So obviously, this is a web application, right? Therefore, we have the user up here, which is interacting with the API gateway. So here, GTScan is basically a search engine for the genome, where researchers would type in the actual gene that they want to target, and GTScan will bring out a ranked list of all the possible options of how to target this gene, so the researcher knows straight away what is the perfect option. So therefore, the input of this API gateway is which gene to target. This gets then deposited into a Lambda function, into a DynamoDB database, and Lambda functions being event-driven. Therefore, the deposit of a certain entry in DynamoDB will trigger the Lambda function. And that first Lambda function is basically a regular expression. You can think of it as sort of a string or a word being searched in a string. And that is the first Lambda function. The results of all of this will be deposited into a second DynamoDB database. So there will be all the possible locations that are there in the genome. From there, you then branch out to these 3 Lambda functions that really identify which one of those sites is the best option. Because as I said, not every site is located equal, and you obviously want to use the best site if you're actually editing precious material like human embryos. So with this being one of the first complicated applications using Lambda functions or the serverless architecture in general, we received a lot of attention. And it's now used by the top research institutions in Australia. And if you want to know more about it, I encourage you to watch the This Is My Architecture video that we recorded last year. So with this, as I said, the reason for going for serverless architecture was because we could not afford anything else. Like when you think about it, the alternative would have been to have a beefy EC2 instance always running so that a researcher maybe eventually interacts with it in order to produce the result, which would have been prohibitively expensive, especially since this is quite a large computation that is in the background. So with this, we obviously wanted to shave off as much excess as possible and keep it as lean as possible. And for that, we needed to identify what are the bottlenecks in our workflow. And for that, we used X-ray. So with X-ray, once it's enabled for your Lambda function, you can record the runtime of each Lambda function. And this is basically what I'm showing here. So these are all the Lambda functions that are included in GTScan. And you can see there are two functions that are particularly offenders in terms of having a super long runtime, particular one. So this is a Perl script, if you know what I mean. Therefore, this is the original architecture that we had, where this little bit basically is that yellow highlight up there, and we have these 2 Lambda functions, which represents these 2 runtimes over here. So in order to eliminate those, we replace them with a new Lambda function. And obviously, being a machine learning group, we replaced it with one Lambda function that had a machine learning, a random forest inside of it. And with that, we were very pleased that we could bring down the runtime dramatically. In fact, we reduced the runtime by 80%. So from 2 minutes to 30 seconds. So as I said, this was, it was doable, but it was quite labor intensive. We had to switch on manually all the X-rays for the Lambda functions, and we had to collect the times individually in order to really find out where's our bottleneck. But there might be a better way. And so this hypothesis-driven architecture, if you want, first you need to encode it as a text. So you don't want to be sitting there manually deploying DynamoDBs or Lambda functions or API gateways. You want to have all of this encoded into a template. And we were using CloudFormation templates or SAM, the SAM config files for Lambda functions. The next step is then to evolve. So for example, if we wanted to compare the 2 Lambda function versus our 1 new Lambda function, we would make a change in the text file to just deploy one and then deploy the other one. But we will still be left with the how to record the actual results of it. And here, Epsagon, which is a startup company in Israel, really had the solution for us. They are specializing on building infrastructure building technology that actually can interrogate your serverless infrastructure automatically. So they figured out a way of injecting something into the Lambda function, and usually, one Lambda function is always connected, or the workflows, the DynamoDB and the rest of API gateways and things like that are typically interconnected by a Lambda function. Typically recording the performance, evaluating whether it was a good or a bad decision, and go around the circle until your architecture becomes better and better and better. So you don't have to rely on hunches of, yes, this might be a better approach to do it, let's test it. Let's test everything and see which one is best. So therefore, I would argue this definitely falls under the DevOps banner, where you want to make continuous changes, but you want to make these changes in a way that is actually manageable. So with that, we do have Variant Spark and we do have GTScan. They're open source technologies, and I really encourage you that if you find this interesting, there's a lot of need for volunteers with the skill sets, the superb skill sets that you have in this particular space. And in fact, this is one of the trends that PricewaterhouseCoopers identified as well, having open source, an open source movement, where basically the IT world or the IT community is coming together, building solutions for one area that can then be applied to a different area, for example, research. So with that, the 3 things I would like you to remember from this talk is, first, datafication will come to your data sets. This is not a threat, but it will come to your data sets as well. Therefore, being prepared for wide data is probably a good recommendation. So I feel this really is a paradigm shift in machine learning to not only think about big data in this dimension, but also think about big data in that dimension. Serverless architecture can be used in a wide range of applications. Specifically, what I was showing here, or was trying to convince you here, was that they can be used for compute-intensive tasks, which was not necessarily what they were designed for, but if you build your architecture in a way to cater or to compensate for that, you can build compute-intensive architecture on a shoestring. Oops. Sorry. And the last one, obviously, is that business and life science research is not that different from each other. In fact, building a community together where your application cases that you have and the solution that you build for those could be applied to life science research or human research, and then ultimately cure cure diseases, which I would argue is, where really the importance lies. So with that, I would like to thank our collaborators and the team and you for listening. Thank you. Do we have questions? We do have time for questions. Okay. Yep, in the back.
Great talk, by the way.
Thank you.
So my question is, you have been focusing a lot on wide data. I'm not very familiar with how medical or genome data comes into the picture, but doesn't the curse of dimensionality play any role?
So the question is, where is the dimensionality in genomic data coming from?
No. I mean that there is this concept where it says that the more number of features you have, the more it is possible that your model will overfit into what you're trying to classify.
Very good, yes. Thank you for that question, definitely. The curse of dimensionality, that's always the elephant in the room, right? And that's the case. Traditional machine learning taught us that you have to have as many samples as you have features. And here we certainly don't have that. We don't have that by an order of magnitude, 2 orders of magnitude actually. And there's no two ways around this. This is not ideal. But we're not getting to a point where we actually will have billions of samples in order to compensate for the millions of features that we have anytime soon, right, to be realistic. Therefore, we need to come up with solutions in the meantime. And this is why we focus on random forests, because random forests are individual decision trees which are weak learners. So these have less propensity of actually overfitting. Well, they have less propensity of being accurate either, but they certainly have less propensity of overfitting. And then only as the ensemble, they retain their inability, is probably a too strong word, but their reduced rate of overfitting, but increasing the accuracy by joining these individual weak classifiers. Gradient boosting tree would be an alternative to that. I think there's less research on how susceptible it is actually to the curse of dimensionality compared to random forest. So this is why we are sticking for the time being with that, because it's a well-known research topic that random forest is probably the best approach to compensate for the curse of dimensionality. But definitely, this is something we always have to be aware of, and that the results that are coming out, the disease genes that we predict, they always need to be conferred by an orthogonal method before we can actually action them. Yes, in the front first.
So how do we train this model with a large set of this genome data if privacy is a concern?
Yes. So the question was privacy and genomic data. How can we actually get the data set sizes that we actually need? And research institutions are actually quite fond of keeping their data in silos. At the same time, they do appreciate and they do understand that data need to be shared in order to get to the right solution. So the question is, how can you marry those two chasms, if you want? And the answer for that is distributed machine learning. So you learn something on your silo, where the data stays in the silo, but the higher abstraction leaves the silo. And you do that over here with that silo as well. And at a third-party trusted advisor that will not have access to the actual data, but will have access to the abstraction, you can join these two things. And if you do that then iteratively, this is how you can learn. It's a fantastic question.
You have told that in genome editing, 7 out of 10 is actually what statistics you got in the success. How the machine learning you are thinking can help in reaching 10 out of 10?
Yes. So obviously we're a long way away from 10 out of 10. But our aim was to enable researchers to interrogate many more scenarios than they could be doing before. So therefore, the speed was important. And the machine learning part to that is that we're predicting, we're able to train a method to say, these particular scenarios, don't bother with them, just focus on these ones. So it's like the lead paralyzation, but in the genome. Does that answer your question?
So actually there are, I think that when you are editing the genome in the embryo, there are two parties there. One is the earlier previous learning, other is the when you are editing, at that time, that is your biological, that medical activity, what you call that editing. You will have some rage or something, by that way you will edit it, right? So at which area you are feeling machine learning will be helpful? It is like targeting the right gene, predicting the right gene, which is actually when you are taking, that is the
Yes,
place?
yes, absolutely. I mean, obviously there's machine learning for the full range, but GT-scan focuses on which gene to target and how to target it.
Okay, so particular chromosome, which place DNA that's where to target, that is based on your machine learning. That's the things you are telling.
Yes,
Okay.
yes.
Okay,
So obviously there's a whole range of biological and molecular aspects to that as well, which is not what we're focusing on here. But ultimately, if we know what the optimal target site is to increase the efficiency slightly, and then keep on that iteration and build data sets on that, with that knowledge, then we can create more data sets to train more methods, to train more machine learning methods in order to keep improving the whole cycle.
Okay. My last part will be probably that in the DNA, see that as I am basically from computer science. So basically, as a DNA, if you are targeting, you should be identify a DNA. So there will be some biological part also will be there, right? So you didn't talk to that about that.
No, that's
okay.
right. So I figured this is not the audience to talk to about
Okay.
biological content. So should we take that offline?
Okay, thank you.
Thank you. I think we'll finish with the Q&A. You could always network with Dr. Denis after we're done.